For a long time, we have relied on flat 2D cell cultures grown on a plate to study cellular and disease mechanisms. It's very simple and cost-effective cell culture process. However, within the last decade, biomedical science shifted to 2D to 3D cell cultures and have become increasingly popular process nowadays because they are more physiologically relevant and better represent in vivo tissues. Hello friends, I am Dr. Manushi Siddarth. In my previous video, I discussed about animal cell culture, how can we perform primary and cell line culture, what are the equipment we need to set up the cell culture lab. So I have entered the playlist of cell culture. Do check it out and you will get a link in description as well as in the I button. In this video, I'll talk about 3D cell culture, why we need it and its assay techniques. So stay tuned at the end and subscribe my channel for more conceptual videos. A 3D cell culture is an artificially created environment in which biological cells are permitted to grow or interact with their surroundings in all three dimensions. Means, unlike 2D environment, a 3D cell culture allows cells in vitro to grow in all directions similar to how they would in vivo. These three-dimension cultures are usually grown in small capsules in which the cells can grow into spheroids or 3D cell colonies. In figure 1, we can see 2D cell culture systems allows the cells to grow in monolayers, whereas in figure 2, 3D cell culture allows cells to grow in all directions, similar to how they would in vivo. Before we go any further details, we should be aware of the terms that are associated with 3D cell culture. Let's start with the extracellular matrix. Extracellular matrix made up of basement membrane and the interstitial matrix. The basement membrane has a mesh-like structure and present between the epithelial cells and the underlying connective tissues. It's strongly cross-linked component of the ECM that made up of sheet-like deposition which is released by epithelial cells. In every given tissue, the basement membrane divides the epithelium from the stroma. It is constantly in contact with the cells, which provides structural support, dividing tissues into compartments and regulating cell behavior. Interstitial matrix contains large amount of collagen or laminin proteins. They also contains glycoproteins like fibronectin, glycosaminoglycans which include heparan sulfate, hyaluronone, proteoglycans and various growth factors like transforming growth factor beta. These matrix also contains other secreted proteins like proteolytic enzymes and protease inhibitors. The fundamental purpose of the extracellular matrix is to aid cell attachment and communication with or adjacent cells. It also plays a role in cell growth, proliferation, motility, and differentiation into other specialized cells and cell signaling. If the component of the ECM are altered, as a result, various diseases such as fibrosis, cancer, and genetic problems would follow. From the delicate tissues of the brain to the hard tissues of the bones, the ECM can have different stiffness and elasticity levels. The ECM elasticity can vary by several orders of magnitude. This feature is primarily influenced by collagen and elastin levels, and it has recently been discovered to play a role in controlling a variety of cell processes. When it comes to tumors, they are frequently stiffer than surrounding healthy cells due to an increase in collagen deposition and cross-linking as well as high hyaluronan acid concentrations. ECM stiffness has also been linked to tumor growth in the past. Physical cues like stiffness often vary in tandem with changes in ECM composition, resulting in bidirectional changes in cells and the ECM. Within a tissue, concentration gradients exist for oxygen, pH, nutrients, effector molecules, as well as cellular metabolites. These natural gradients are influenced by the proximity to blood vessels by the diffusion of molecules through the ECM. Yes, ECM is important to exchange or to regulate the oxygen and nutrient consumption with the cells and the production of cellular waste products also. Molecular concentration gradient affect various cell behavior including cell motility, cell migration and cell signaling, and are important in chemotaxis and morphogenesis in normal development and in wound healing. If we talked about tumor, 3D tumor spheroids are much more representative of in vivo tumors. There is peripheral layer having proliferative cells due to the closeness to the supply of oxygen and nutrients, underlaid by an intermediate layer having quiescent cells and an inner necrotic core. This sphere can mimic inward diffusion to form oxygen and nutrient gradients and a perfect model for in and outward diffusion of regulatory molecules and provide a reservoir for the accumulation of waste. Another thing we should know that in vivo cells are not only surrounded by ECM and ECM associated signaling molecules, but connective tissues also contain stromal cells, including mesenchymal supporting cells such as fibroblasts or adipocytes in epithelial tissues, glial cells in neuronal tissues, cells of the surrounding vasculature, and cells of the immune systems. Interaction with stromal cells can regulate surrounding epithelial and neuronal tissue, which contribute to disease progression and influence the therapeutic response of the cell. In living tissues, cells exist in 3D macro environments with intricate cell-cell and cell matrix interaction and complex transport dynamics for nutrients, oxygen, soluble factor, and cells. Standard 2D or monolayer cell cultures are inadequate representation of this environment, which often makes them unreliable predictors of in vivo drug efficacy and toxicity. 3D spheroids more closely resemble in vivo tissues in terms of cellular communication and the development of extracellular matrix. These matrices help the cells to be able to move within their spheroid, similar to the way cells would move in living tissues. The spheroid are thus improved models for cell migration, differentiation, survival, and growth. Now let's compare the 3D cell culture with 2D cell culture. In 3D cell culture system, cells will allow to grow in 3D environment where cell-cell and cell matrix interaction occurs, whereas in 2D cell culture system, cell-cell interaction is restricted and cell matrix interaction is absent. In 3D cell culture, there is a gradient for nutrients, oxygen, pH and others as we talked previously. In 2D cell culture, gradient mechanism is totally absent. In 2D cell culture system, cells exposed to liquid interface, whereas in 3D cell culture system, cells are embedded. It's now evident why we need 3D cell culture. If you think about it, no cell types within our body grow as a monolayer or independent of other cells or tissues. Instead, most cells naturally exist in complex 3D structures, including different cell types within an extracellular matrix. The numerous cell-cell and cell matrix interaction all have a profound effect on their behavior, and there is a possibility to co-culture two or more different cell types. In 2D monolayer, there is a uniform access to nutrients and oxygen, which is not the case in tumors. As we talked about in previous slides, 3D tumor spheroids are much more representative of in vivo tumors, in which inner cells have less access to nutrients and oxygen compared to the outer layer, because they forming a natural gradient. Now let's talk about the 3D cell culture models. They have been divided into three categories. First is anchorage independent, that is also called non-scaffold based. Second is anchorage dependent, also called scaffold based. And third is specialized 3D culture platforms. Anchorage independent includes two techniques. First is hanging drop and another is low attachment plates. Anchorage dependent, which is scaffold based, includes hydrogel technique, which is divided into biological and synthetic hydrogels. Specialized 3D culture platforms includes macrofluidic devices and micro patterned plates. So now let's learn these techniques in details. Scaffold based culture technologies provide physical support ranging from simple mechanical structures to ECM-like matrices on which cells can aggregate, proliferate, and migrate. In scaffold-based 3D cultures, cells are embedded into the matrix, and the chemical and physical properties of the scaffold material will influence cell characteristics. Scaffold can be of biological origin or they can be synthetic engineered to mimic key properties of ECM, such as stiffness, charge, or adhesive moieties. In some synthetic scaffolds, growth factors, hormones, or other biologically active molecules can be encapsulated to enhance cell proliferation or to promote a specific cell phenotype. During 3D cell culture scaffold selection for a certain application, you must consider the physical as well as biological properties. Physical properties includes porosity, stiffness, and stability of scaffold in culture, and biological properties defined cell compatibility or adhesiveness. Hard polymers can provide the physical support found in a specialized tissues such as skin, tendons, or bone, and micro-patterned surface microplates can be designed for specific application such as support of cell networking. Hydrogels are networks formed from dilute polymer chains with given structure and properties, obtained either by intermolecular or by interfibrillar crosslinks. Although hydrogels display solid-like material properties in the quiescent state with over 95% of water by volume, they can indeed provide a cell-liquid interface. Hydrogels may come from natural sources or can be synthetic, with the possibility of mixing different hydrogel materials to obtain hybrid hydrogels possessing new physical and biological properties. Hydrogels from natural sources such as collagen, matrigel are biocompatible, have natural adhesive properties, and sustain many physiological cell functions, resulting in high cell viability, controlled proliferation, or controlled differentiation, and often a cell phenotype typically observed in vivo. Collagen, with type 1 collagen being the most abundant form, is the most widely used ECM protein for 3D cell culture. Gelatin is a partial thermally and chemically degraded product of collagen and can be stabilized by covalent modification. The possibility of covalent linking of functional groups also allows for the production of specialized gelatin gels. Another natural hydrogel is alginate, that is isolated from the cell walls of brown algae. The mechanical properties and rapid degradation of the alginate hydrogel somewhat limits its application for 3D cultures, but alginate hydrogels have found their use as a 3D printed scaffolds for specialized tissues such as vascular tissue, bone, and cartilage. When well designed, synthetic hydrogels are ideal material to use as 3D cell culture scaffold as they can mimic biological properties of ECM and be functionalized with defined adhesive moieties that include proteolytic sites and encapsulate growth factors. At the same time, they are chemically and physically well defined and often have tunable mechanical properties to achieve a desired stiffness or porosity. Synthetic hydrogels can be categorized into non-natural and natural polymers. Polyethylene glycol, polylactic acid, and polyglycolic acid and other unnatural polymer hydrogels have the advantage of being comparatively inexpensive or relatively inert and have reproducible material properties that are usually easy to tune through synthesis or cross-linking and are more reproducible, thereby supporting the acquisition of consistent results. On the other side, unnatural polymers lack adhesive moieties found in natural ECM and require cross-linking of biological peptide to the scaffold to improve functionality. Polyethylene glycol gels and their derivatives have been used in a variety of 3D cell culture applications, including stem cell differentiation, cell invasion, and angiogenesis. Synthetic natural polymers share with non-natural polymers the advantage of consistent and tunable material properties for reproducible results. Due to the biological nature of their naturally occurring moieties, natural synthesized hydrogels are highly compatible with encapsulation of cells for 3D cell culture. However, material cost can be high due to complex chemical synthesis requirement. One of the best characterized natural hydrogel is high hyaluronic acid, a glycosaminoglycan that can be modified with functional groups, allowing for the formation of hydrogel with diverse properties for a wide range of applications. Small peptide-based hydrogel material are an evolving field in materials science and provide a new 3D cell culture technology. 3D cultures can be grown without the presence of a supporting scaffold, and that includes anchorage independent technologies. Scaffold free methods depend on cells to self assemble into clusters or spheroids. These spheroids are grown in specialized culture plates, which includes hanging drop microplates and low adhesion plates with ultra low attachment coating. These spheroids can recapitulate physiological characteristics of tissues and tumors with regard to cell-cell contact, and they can synthesizing their own ECM, which can allow for natural cell-cell matrix interactions. Spheroid size depends on the initial number of cells seeded, and spheroids can be grown to a size where they can display oxygen and nutrient gradients similar to tissues. Hanging drop cultures are a well-known 3D culture technology taking advantage of cell aggregation of cells into spheroids when a cell surface is not available for cell attachment. Hanging drops can be created in specialized plates with open, bottomless wells that are designed for the formation of a small droplet of media. The, the droplet is big enough for cells suspended in the medium to aggregate but small enough to prevent it from being dislodged during manipulations. Hanging drop cultures have also found applications in toxicity testing in hepatocytes. In a recent study, human primary or induced pluripotent stem cells derived cardiomyocytes were co-cultured with endothelial cells and fibroblasts in a ratio similar to that found in vivo. The cardiac spheroid mimicked important in vivo features of the human heart biochemically or pharmacologically, offering a 3D cell culture model to study toxic effects in human heart tissues. Similar to hanging drop plate cultures, spheroid macroplates with round tapered or V-shaped bottoms take advantage of the lack of cell attachment surfaces to promote aggregation of cells and spheroid formation. In contrast to hanging drop plate cultures, a transfer of spheroids to a different plate for prolonged culture or experimental procedure is often not necessary. Low adhesion plates are often made of polystyrene and treated with hydrophilic or hydrophobic coatings like the non-adherent polymer poly-HEMA or natural polymers such as agarose. The coating reduces the attachment of cells to the point that they preferably aggregate with each other to form spheroids. Because of their larger volume, low adhesion plates are well suited for multicellular culture and are frequently used for studies in tumor cells. Magnetic cell levitation is an emerging technique for the formation of spheroids. To generate spheroids, cells are preloaded with magnetic nanoparticles and then, using an externally applied magnetic field, they are floated toward the air liquid interface within a low adhesion plate to promote cell cell aggregation or spheroid formation. Magnetic levitation has been used to generate spheroids from cells of various tissues to form multicellular mesenchymal stem cells spheroid and for tissue engineering. Microfabrication technology allows for the fabrication of an endless array of imprinted micropatterns on the surface of plates. The micropattern plates are designed to promote cell-to-cell adhesion for scaffold-free macrosphere formation within the confinement of a macrospace. These plates are coated for low adhesion. Micropattern plates can also be manufactured to provide a scaffold-based 3D culture environment to promote cell attachment for the formation of contagious networks along surfaces and organoids. Polymeric prefabricated scaffold such as porous disc, electrospun scaffold or orthogonally layered polymers are physical supports that can be an inert matrix or designed to mimic in vivo ECM on which cells can attach, migrate or fill scaffold compartments to form 3D cultures carrying a geometric configuration. Currently, the most common application for such scaffolds are for tissue regeneration, recreating the natural physical and structural environment of bone, ligaments, and cartilage for skin, vascular, skeletal muscles, or CNS tissue, and for preclinical in vitro 3D culture testing for tumoroids or engineered tissues, like organoids. On the other hand, microfluidic devices are designed for cell cultures under perfusion and allow for steady supplies of oxygen and nutrients while at the same time removing waste. Macrofluidic devices can be built to mimic shear forces found in vivo in cells that are exposed to blood flow like endothelial cells. A barrier between compartments can be physically incorporated into the device, or it can consist of a non-physical barrier such as a supporting matrix mimicking ECM. Microengineering of microfluidic devices also allows for the development of organ-on-a-chip platforms with 3D tissue models have been described for a variety of organs including skin, muscle, liver and neural tissues, bridging in vitro cell culture and in vivo animal models. With the advancement of these 3D culture technologies, organ-on-a-chip are poised to provide advanced tools for drug development and high throughput screening in the future. This is a references slide. So thank you for listening. For upcoming videos, don't forget to subscribe and give thumbs up if you like this video. Stay tuned. Take care.